Okay, welcome to the second podcast in the 48 series, where the first one we looked at an overall introduction to photosynthesis. In this one, we are going to look at the details of the first set of chemical reactions that allow photosynthesis to occur. There are two sets. This will be set one. These are referred to as the light reactions of photosynthesis. Let me just get into our slides, shall I? All right. Just like cell respiration had three sets of biochemical pathways, or three biochemical pathways, the glycolysis, the Krebs cycle, and the electron transport chain. Photosynthesis has two biochemical pathways that allow plants to create sugar molecules. Okay? So two sets of reactions, and they are often called either the light reactions and the dark reactions. That's really, the light reactions is a fine name. The light reactions require light. However, the dark reactions do not require dark. It's just that they don't directly require light in order to occur. However, these reactions can't occur if these reactions don't occur, and these reactions do require light. So at any rate, the bottom line is this is not the desirable name for the second set of reactions. This is a fine set of names, the light-dependent reactions and the light-independent reactions, meaning these reactions, again, are like they depend on light, and these don't depend on light. What we will tend to use is this pair of names. Remember, these are all the same processes, just different names. The light reactions and the Calvin cycle. That's the second set of reactions to do photosynthesis. Okay? Fundamentally, the light reactions must have light in order to proceed. The Calvin, do you see this? The Calvin cycle can occur without light. We've already talked about that. Okay? But the Calvin cycle cannot happen if the light reactions have not happened. Basically, the Calvin cycle still is dependent on light in some form or another. All right, so the light reactions are basically there to convert light energy, which is not a very usable form for living things. Light energy, living things cannot use it very well. We are going to convert it to a chemical form of energy that living things can use. We are going to convert it to essentially ATP and another molecule called NADPH. Okay? Now, the thing about these two forms of energy is that they are very temporary. They're unstable forms of energy. They don't last long. So this is kind of just a middleman that is going to get us to where we want to be in photosynthesis, and that is to the formation of glucose molecules. Okay? So in the light reactions, we are going to be doing this step here. We are converting that light energy and capturing it in a chemical form. It's just not a very adequate chemical form for us, at least not yet. Okay? So we're converting solar energy to chemical energy, which is going to be ATP and our NADPH. Remember, we dealt with a similar electron carrier back in cell respiration. It was NAD. Okay? The Calvin cycle then is going to take these things here and convert them to glucose. Okay? So those are, the Calvin cycle is essentially the sugar building reactions where we are going to reduce carbon dioxide that the plant takes in from the atmosphere. We're going to reduce it, meaning we're going to add electrons to it, and when we do so, we're going to build this complicated glucose molecule. Okay? So We saw this slide previously in our introduction. We're going from light essentially to ATP, ATP and NADPH, and then finally to glucose. And we need these raw materials in order, these raw materials here in order to do that. Okay. Light reactions begin, well, let me take a little step back here before we go. Remember that the light reactions begin in a chloroplast, okay? And a chloroplast is a double membrane structure, probably was originally a free-living bacteria, became engaged in a symbiotic relationship with a larger cell. But this chloroplast, or organelle, has in it little stacks of structures, membrane-bound structures called thylakoids. The stacks are called grana, or granum, singular. Okay? So the activity of photosynthesis takes place on one of these thylakoids, every one of those thylakoids. And it is specifically in the membranes because there are proteins embedded in these membranes of the thylakoids. Now, the first protein that we are going to talk about that is embedded in these thylakoid membranes is called a photosystem. Okay? It is a photosystem is a complex that has chlorophyll in it, but not just chlorophyll. It's got the other pigments as well that are all there. All these photosynthetic pigments are there to help catch light, okay? Embedded in the thylakoid membrane. Now, the photosystem has, if this is my photosystem, it's a protein embedded in the thylakoid membrane, so there's the thylakoid membrane. Embedded in the top are a bunch of chlorophyll and other pigment molecules, carotenoids, whatever. This region here is called the antenna complex. You need to think of it as a catcher's mitt there to catch as much and as many photons of light, units of light, as we can. Okay? Much better picture than the one I just drew, eh? Alrighty. Let me delete my chicken scratch. There we go. Alright, this is a much better picture. This region is the antenna complex. It's sitting on top of the protein glob we call a photosystem. Okay? Indicated there. When a photon manages to hit one of the chlorophyll molecules or other pigment molecules in the antenna complex, the energy from that bounces around randomly on the chlorophyll molecules until that energy strikes a very particular chlorophyll A. Remember, chlorophyll A is the special chlorophyll that engages in photosynthesis for us. The others are helpful in other ways, chlorophyll B, carotenoid pigments, and so on, but chlorophyll A is the one we really care about. Okay, so we've been over all that. So, and it lands on that particular chlorophyll A molecule. Now, once that energy has landed on this chlorophyll A molecule, it excites its electrons. It excites a pair of electrons to a higher energy level. Okay, which is typical. Light energy, if it's enough light energy, will excite electrons in any atom to a higher energy level. It's just that normally those electrons, just as though you threw a ball in the air, those electrons will eventually just fall back down to their original energy level. However, in the antenna complex, in this region right here, I'm highlighting with this square, there is a region we call the reaction center. Okay? In that reaction center is the very special chlorophyll A molecule that is going to contribute its electrons, and something called the primary electron acceptor. When these electrons get excited, instead of falling back down, they are caught at their higher energy level by the primary electron acceptor. So this catches the excited electrons, okay? And that at that point we have captured the energy that was in this photon that bounced around, excited these electrons, and now we have caught those electrons at an elevated energy level. That's critical. That at that point we have converted this light energy to a chemical energy form. Okay? It's not particularly useful to us yet. We've got to go through some steps before we're going to make glucose with that energy, but we're on our way. All right? We talked about, let me see something. Yes, we talked about this last time. This is where I wanted to be. I just want you to realize how similar hemoglobin and chlorophyll look. This is chlorophyll. This is hemoglobin. They are both pigment molecules. This is a green pigment for photosynthesis. This pigment is red, and it's found in your blood cells, and it carries oxygen. But both of them have atoms, inorganic metal type atoms in the center. This one's got magnesium, this one's got iron, but they look really similar. They behave, they absorb particular wavelengths of light and reflect others. So I want you to be able to at least recognize these molecules for what they are. Recognize that if you see magnesium in the middle of one or iron in the middle of something that looks like this, what you're dealing with, it's very helpful, okay, in terms of the AP test. All right, so that reaction center, which we've talked about, and the primary electron acceptor, it's going to catch those high energy electrons. Here we are, another slide image just of the electrons getting excited into their excited state. Normally they would fall right back down again, but in a chloroplast there is that primary electron acceptor that will catch them and hold them at that elevated state. Okay? We have now stored that energy in a chemical form. Very important. All right, there are two of these photosystems in photosynthesis. We call them photosystem 2 and we call them photosystem 1. Photosystem 2 comes first. It was discovered second, but it comes first in the chain of reaction. It best absorbs light at a wavelength of 680 nanometers, so sometimes you'll see photosystem 2 referred to as P680. It's just another name for photosystem 2. This photosystem best absorbs light at a wavelength of 700 nanometers, so it's called P700, just another name for photosystem 1. Okay, you can see that chlorophyll B is the pigment at work there. Chlorophyll A is the one you need to know. Okay, this is the one you really need to know. Okay, all right. We are going to talk about non-cyclic electron flow. In other words, if this is my thylakoid and I start at photosystem 2, which comes first, I'm going to move my electrons from that all the way to some endpoint in a very linear path. All right? There is another type of flow called cyclic electron flow, where when the electrons are flowing from the chlorophyll, they will stop and kind of loop around in a loop for a little bit. We'll talk about that second, okay? All right. Please be aware that oxidation reduction reactions are going to play a role in passing the electrons from one chemical group to the next, just like they did in cell respiration. Okay? Electrons move from one thing to another by oxidation reduction reactions, because one compound will be more electronegative or electron loving than another, and so the electrons will move to the one that loves them the most. Okay? So here's our steps. Non-cyclic linear electron flow. First thing that happens, we have a photon that strikes the antenna complex at photosystem 2. This is going to excite, the energy will bounce around the antenna complex, but eventually it will land on the very particular chlorophyll A in the reaction center. and then those electrons will be excited and be caught by the primary electron acceptor, which is being indicated right there. So we're catching the electrons at an elevated energy state. Okay? Now, this leaves behind an electron hole in that photosystem because these electrons are now moving off, and the chlorophyll A that donated them has no electrons. Okay? In order to replace them, a molecule of water is going to be broken down by an enzyme. The water is in the stroma, the surrounding fluid that surrounds the thylakoids in the chloroplast, and it is going to extract electrons from the water molecule and donate them to the chlorophyll A. Okay, so the water molecule is essentially going to be split. The hydrogen ions will be used. We'll see what that is in a minute. And the oxygens that remain will combine together to form oxygen gas, which will be released as kind of a waste product of photosynthesis. Okay, but this is why water is needed for photosynthesis. If you'll remember, the photosynthetic reaction, of course, is CO2 plus H2O plus light is going to give us glucose and oxygen. We need water to replace the electrons from the chlorophyll A after they get excited and leave the chlorophyll A. Okay? It is also where the oxygen gas comes from. Oxygen gas is coming directly from the breaking apart of the water molecule to extract electrons, and the oxygen gas comes from that water molecule, or that water molecule and others. Okay? So we're going to take the electrons and pass them from that acceptor. In photosystem 2, we're trying to get them to photosystem 1. But there are stepping stones in between those two things. If this is my thylakoid membrane here, photosystem 2 is here, photosystem 1 is here, but we are going to have some stepping stones to help us get the electrons over to here. That is an electron transport chain, very much like what is in the mitochondrion. Okay? As the electrons are passed along the electron transport chain, they lose a little bit of energy. But just like in cell respiration, they don't just lose energy to the ether, to the vacuum of space. That energy is used to do something. It is used to pump protons from the stroma. Out here, there's protons. Remember, we broke down water, so there's going to be lots and lots of protons out here. We're going to pump those into the thylakoid space, the thylakoid compartment inside the thylakoid. Okay? So we are creating basically a proton gradient where we will have a very high concentration of protons inside and thus a lower concentration outside. Okay? Naturally, those hydrogen ions want to flow with their concentration gradient. They will do so only where they are allowed. And so there will be a molecule of ATP synthase that will allow hydrogen ions to fall through it with their concentration gradient and will create ATP as a byproduct or as a product of that. Okay? So ATP synthase is again an important thing. Okay? So this is a nice diagram of everything we just talked about. We see in this diagram the light hitting the antenna complex, the energy being bounced around until it lands on the particular chlorophyll A in the reaction center. Those electrons get excited, and before they can fall back down, the primary electron acceptor catches them. But because this electron transport chain is more electronegative than the electron acceptor, the electrons begin a journey from here to there to there. Each of these is more electronegative than the one that came before it. We will pump hydrogen ions from the stroma through these proton pumps into the thylakoid compartment. Where did these come from? They came from the splitting of water. Here's a water molecule. We break it apart. We get hydrogen ions. Why did we break apart the water molecule? Because we needed electrons to replace these that are now journeying on this way. So the water molecule gives us electrons, it gives us hydrogen ions over here, and we get oxygen gas as a waste product. And we are making ATP as the hydrogen ions that are pumped into the thylakoid compartment fall through ATP synthase. Okay? All right. Now, the rest of this process, okay, when we use ATP synthase to make ATP, please remember this term. It's called chemiosmosis. Okay, we're using that proton gradient to allow H pluses to fall through ATP synthase and create ATP. That's chemiosmosis. Now, when we use light energy ultimately to drive this type of ATP synthesis, we essentially call this chemiosmosis, we call it photophosphorylation, because we are using light to stick a phosphate group onto an ADP molecule. Okay? So we used terms earlier in the year, substrate level phosphorylation, we've used oxidative phosphorylation. Well, this is kind of like similar to this, only we're not using oxygen to drive the phosphorylation here, we're using light, so we call it photophosphorylation. Okay? All right. Now, as we move on, the electrons, when they reach the end of the electron transport chain, they are now ready to become part of photosystem 1. Okay? In photosystem 1, light also strikes photosystem 1. If light's striking over here, no doubt it's striking over here as well. Same procedure happens. Electrons from this chlorophyll A get excited. They are caught by a primary electron acceptor and they are sent on their way. But as these electrons that originally came from photosystem 2 fall down their electron transport chain, okay, they can jump into the hole that was left by these electrons and fill it up. So this chlorophyll A will be ready to go again. How is this hole going to get filled up? As these electrons jump in and fill that hole, this electron hole gets filled up from these electrons that came from the water molecule. Okay? So, all right. So, the electrons that come from photosystem 1 are also going to be sent on their way, okay? And they are going to be passed to a compound called NADP plus reductase. Let me get to my slide. Here it is. NADP plus reductase. So the electrons here are going to get passed to NADP plus reductase, where a molecule, it's a coenzyme, it's an electron carrier, is going to come pick up these electrons. When it does so, it will be called NADPH, and it's going to carry those electrons and, most importantly, their energy away in order to do something. Okay? So, NADP plus reductase is the enzyme embedded in the thylakoid. NADP plus is the coenzyme, the little taxi cab. It's going to drive up to the enzyme here and it's going to pick up its electrons that it's going to carry. When it does so, it becomes NADPH, and then NADPH is going to carry these electrons to the place where sugar will be made, and that's the Calvin cycle. Okay, we are going to use their electron energy to make sugar. We are also going to use the ATP we were making in the light reactions as well. Okay, so we're going to use this and this to make sugar. Okay, alright, so the light reactions, the bottom line is those light reactions are going to result in ATP and NADPH. Both of them are essentially energy sources. They are going to provide the energy we need to make glucose. Okay? Making glucose involves catching CO2. CO2 is our carbon. Remember, glucose is C6H12O6. Well, we got to get carbon from somewhere. We are getting it from CO2, from the air. It's going to take energy to force carbons and hydrogens and so on together to build a complex glucose molecule. This is where that energy is coming from. Okay? So, without this chemical energy we get in the light reactions, the so-called dark reactions, the Calvin cycle, wouldn't happen. Alright, do remember that we talked in class about how you need more ATP than you do NADPH for the Calvin cycle, so that we need to run the ATP making mechanism more. So there is something called cyclic photophosphorylation, cyclic electron flow, where instead of the electrons, if this is photosystem 2 and this is photosystem 1, the electrons get excited, they pass here, and they ultimately linear. Instead of doing that, the electrons get to here and they double back and they keep going down this little water wheel, and that going down there causes these proton pumps to work, and that increases the production ultimately of ATP. So this makes lots of ATP but does not result in any NADPH. That's because the Calvin cycle needs more ATP than it does NADPH. So that's the simple reason for that. Alright. Okay, so that's just kind of showing you how much more ATP is made than NADPH needed to make a glucose molecule. We need 18 ATPs and 12 NADPHs to make one glucose, so we've got to run that ATP mechanism a little more often. Okay? Alright, so photophosphorylation, making ATP using light, and that's this part of the light reactions. This part does not make ATP. This part over here is the NADPH making part. Okay? Alright. The cyclic phosphorylation will happen in here when it happens. Yeah, like that. Okay, we'd get to this photosystem 1 and then double back and fall down the electron transport chain again. Okay, so summary. Where did the energy come from? The energy in the light reactions comes from light. Where did the electrons come from? They came from water. Where did the water come from? Well, that came from the soil and was transported up to the leaves and the xylem. Where did the oxygen come from? The oxygen came from the breakdown of water. Where does the oxygen go? It goes out into the air. Thank goodness we're breathing it. Okay? Where do hydrogen ions come from that fuel ATP manufacturing? That came from water as well. Where does ATP come from? It comes from the activities of the light reaction, particularly the cyclic part where we are running that electron transport chain. Okay? I don't know why my pen keeps turning off. ATP will be used to provide the energy for making glucose in the Calvin cycle. Alright, where does NADPH come from? It comes as the end product of the non-cyclic or linear photophosphorylation or electron flow. It comes from that, from the activities of NADPH, NADP plus and the NADP plus reductase, and it also will be used for making glucose in the Calvin cycle. Okay, so we will now talk about the Calvin cycle. We're done with this. Let me know if you've got any questions. See you next time.